Yeah, hi. Uh, Hi, Hamza. How are you today?
Yeah, I'm good. Thanks.
Okay. Are you ready to start the interview?
Yeah, that's fine.
Okay. So the first question, can you see the graph here?
Yeah, yeah,
Yeah,
I
is
can.
that clear? Right. Can you describe the graph to me, please?
Okay, so yeah, hi, I'm Hamza. And so this graph, it is, so there's an initial kind of rapid increase in the oxygen saturation
Mhm.
um, when the partial pressure increases, and then that ends up plateauing. Um, so there is an overall sort of S shape or sigmoid shape
Mhm.
to the graph.
Okay. And why is the graph this shape?
So I think it's to do with oxygen binding to the hemoglobin. And what it's showing is it's showing that at low partial pressures of oxygen, a slight increase in that oxygen level will lead to a large increase in oxyhemoglobin present. And this is because of the fact that it's much easier for oxygen to bind to the hemoglobin molecule. Um, but by the time it gets to the high partial pressures, because most of the oxygen binding sites will be full, it's going to make it slightly more difficult. So there's going to be that restriction where it can't get to that 100% level as easily at the high levels.
Yeah, okay. So why would you say that this is advantageous for humans to have this with hemoglobin?
Well, um, I guess it means that at low partial pressures of oxygen, which are found in sort of the actively respiring tissues that are going to need the oxygen, it means that the oxygen will um, dissociate from the hemoglobin and go and provide itself to the tissues that are going to need the oxygen for respiration.
Mhm.
Whereas, but it also means that at really high partial pressures of oxygen, even small decreases aren't going to make a huge difference. And that's why we have a flat upper portion. So it means that it takes a significant lack of oxygen to start having a change in the body's ability to kind of take up oxygen and use it.
Okay, yeah. Um, so how would you describe or draw the effect of carbon dioxide on the shape of the graph?
Uh, I think with carbon dioxide, there's a shift to the curve will move sort of to the right because what happens is that carbon dioxide then competes with oxygen for the hemoglobin, and because carbon dioxide has a higher affinity to oxygen to the hemoglobin than oxygen, it means that the carbon dioxide will bind more strongly to it. I guess this is more helpful in respiring tissues because it means that, again, when there's high carbon dioxide in respiring tissues, more oxygen is dissociated and so that provides more oxygen for further respiration.
And do you know the name of what you're describing?
Yeah, I think that's the Bohr effect.
Mhm. Okay. How might this graph that you see be different in a fetus?
Let me think. So I know that in a fetus, obviously, they take their oxygen from the maternal red blood cells because they don't have lungs to actively respire themselves or to actively inspire. So the hemoglobin is going to need to have a higher affinity for oxygen. So I think I'm going to guess that it probably shifts to the left um, because it means that at the same partial pressure of oxygen, there is a higher affinity in fetal hemoglobin, so that the oxygen dissociates from the maternal red blood cells and donates itself to the fetal red blood cells, and that means that oxygen then can be taken up for the fetus, for them to do respiration for growth.
What other factors affect the oxygen carrying capacity of blood?
So I think obviously the amount of red blood cells, because if you have less red blood cells, then you can't carry as much oxygen. What else could there be? Oxygen maybe that's dissolved, because that might affect like the concentration gradients and diffusion ability from the lungs to the blood. Yeah, I think that would be those would be the core ones.
Okay, lovely, thank you. So we can take a few seconds before we start the next scenario, is that okay?
Okay. Yeah, that's fine.
Hi, Hamza. Hi, Hamza. So welcome to the second scenario. Are you ready to start?
Yeah, that's fine.
So the first question is, can you see my screen okay? Yeah.
Yeah, I can.
Yep. The
Thanks.
first question is, how do vaccines work?
So I'm not quite certain about this, but so I know that the goal of a vaccine is to provide like long-term immunity against a pathogen without having developed those signs and symptoms originally. And I know that like some vaccines kind of they inject like a dead or attenuated form of the pathogen, and that like that will do something, but I'm not exactly certain like how it gets from like the kind of injection to the long-term immunity.
Okay. Is there any type of cell that you think helps with the immune response once you've been given the vaccine?
So I guess if we're just like thinking about normal kind of immunity when you're exposed to an antigen, maybe antigen presenting cells that might like macrophages that might come along and like do phagocytosis, which makes them into antigen presenting cells. And then maybe from there, maybe you have activation of T cells potentially, and then they'll activate B cells, which will produce maybe memory cells. But is that kind of, am I on the right lines there?
Well done. Yeah, we'll move on to the next question. Can you name any types of vaccine?
So I think, yeah, I think you've got the dead dead pathogen vaccines. It's like an inactivated vaccine. I guess you'll have also like it'll be an live, but it'll be attenuated, so it'll be seriously weakened, that it won't cause severe symptoms. And I think that would be something like, I don't know, I think like maybe the COVID vaccine might be live or attenuated form. And then yeah, I think those would be the main ones I'm aware of.
Okay, we'll move on to the next question. What are the risks that you know of live vaccines?
So I guess like any drug or any treatment, you could have severe allergic reactions to it. And yeah, also if there is like a vaccine that isn't properly attenuated, it could cause the disease then, which would be a problem, especially for serious infections. There could also be that virus could like mutate and become worse or more deadly form of it, and then that could then cause further issues.
Okay. Why are live vaccines more effective?
Is it because they they are like constantly triggering that immune response. So like it becomes there's a more effective and efficient immune response that's been triggered from it. So maybe that would be helpful to developing longer immunity, but I'm not exactly sure again how that happens.
Okay. What type of vaccines do you know are being used for COVID?
So I've heard like there's a lot of like mRNA based vaccines, which are like the new kind of type. And I know that mRNA is the molecule that is used in transcription or made in transcription, and it's used to guide translation in protein synthesis. So maybe what the mRNA vaccine is doing is it helps to kind of prevent that kind of it maybe creates molecules that will prevent transcription of viral proteins.
And do you know any other?
Not really sure.
No problem. Okay. Okay. We're ready to move on to the next question.
Yeah.
How would you design an ethical study to test how effective a vaccine was?
So I guess like any good clinical study, it would have to be double-blinded with a placebo control, and there would be randomization of the groups. Obviously, the problem that there might be with that is that there might be an ethical problem. If some people get the disease and get seriously ill, that would not be a good thing. Or the alternative is to do like with a lot of like I don't know what the term is, but they're like studies that kind of after the event has happened, and you see like what percentage did it work or did it not work with the natural infection.
Okay. Can you think of any viruses or other infectious disease that we don't have a vaccine for yet, and suggest why this might be?
So malaria, we don't have a vaccine for. I think, well, firstly, because it's a parasitic infection, the natural infection does not elicit a proper immune response. And also, maybe there's not enough funding for it at the moment. HIV, which I think is quite difficult to do a vaccine for because it targets the immune cells themselves. So that then becomes like it's almost like it's difficult because if you get a vaccine for it, it's targeting the immune cells that you're trying to make against it. And also there isn't a lot of antibody response to it. So I think those might be kind of the key ones.
Okay, lovely, thank you. So we'll just take a few seconds before we move on to scenario 3. Is that okay?
Yeah.
Okay.
Okay.
Okay. So we're on to scenario 3 now on antibiotics. Can you see this diagram clearly? I can zoom in a little bit more.
Yeah.
Okay. Can you see what the arrow labeled 1 is pointing at? So this arrow here.
So I think that's like after the incubation, there's where the bacteria are present within there, because obviously the agar plate will contain all the nutrients required for the bacteria to grow, and the incubation will provide the right temperature for it to promote that growth. then arrow 1 points to where the bacteria is growing.
Yeah, okay. We'll move on to the next question now. What is the arrow labeled 2 pointing at?
So that's going to be the opposite of that. So that'll be the zone of inhibition, where the antibiotic has prevented the bacterial growth. And it's basically the antibiotic is killing the bacteria by preventing growth, division, and replication.
Okay, great. So that was the answer to the next question. So we'll move on to the question after that. Which of these is the best antibiotic and why?
So the whole principle to this is like the idea that the larger the zone of inhibition, the more bacteria have been prevented from growing. So we're looking for the antibiotic with the largest zone of inhibition. Therefore, it's going to be C.
Okay. What makes a successful antibiotic?
One that's quite targeted to a specific bacteria and not the host. One that will target a specific function that the bacteria needs to replicate and survive. So one that is ideally one that kills the bacteria as well as preventing its growth. One that doesn't affect host bacteria, and one that doesn't gain resistance quite easily would be quite good, because obviously antibiotic resistance is a big problem.
Why is there no zone of inhibition around A?
Maybe, well, first of all, it could be a placebo, so like a negative control where you have like a saline solution or some other type of effect that isn't going to affect it, or it could be an antibiotic that the bacteria has already developed resistance to, and so therefore cannot inhibit its growth.
Okay. Move it down here. What do you understand by the term antimicrobial resistance?
So antimicrobial resistance is where the bacteria is no longer susceptible, or it can't be affected by a particular antibiotic. And this is done by the process of natural selection. So the presence of the antibiotic leads to the development of resistance genes, which then further lead to more selection of that specific gene until the colony is fully resistant.
Okay. And final question for this scenario. How can we combat antimicrobial resistance?
So we can do that by decreasing the use of antibiotics, making sure the antibiotics that we use are selective to the disease that we need them to be effective against, developing new antibiotics, but obviously that's going to be a slow process, and it's going to be a bit slower than the actual process of antibiotic resistance developing, making sure patients finish their antibiotic course, because I think that there's a lot of problem with that. Yeah.
Okay, thank you very much. So we'll take a few seconds before we start the next scenario. Is that okay?
Yeah, that's fine.
Okay. So we can move on to scenario. So we're on to scenario 4 now, bodily fluid. How could you measure the volume of blood in the body?
I guess you could do it by measuring how long it takes for something to kind of pass through the body, and if you know how much like is filtered by the kidney in a particular amount of time, measuring how long it takes for the concentration of a specific substance to kind of decrease by a certain amount, and doing that by measuring how long it takes to pass through the body will tell you how much it's been dissolved in, kind of thing. Is that kind of the right lines?
Okay, moving on to the next question. Okay, you injected 150 milligrams of sucrose and leave to mix over a few hours. After this, the plasma sucrose levels are 0.01 milligrams per milliliter and 10 milligrams are lost due to metabolism. Work out the volume of distribution. I'll leave that up there so you can see.
I'm not quite sure what the volume of distribution is. Is there like a definition for that?
The definition is the equation itself.
Okay.
That you would use.
So is that, I guess if I'm just gonna like try and break it down as a word, the volume of distribution is kind of the amount that the substance will need to be spread out in. And so that would then be the how much you've injected minus how much you've lost, obviously, divided by how much is left after a few hours. So then you just do 150 minus 10 divided by 0.01. So that's 140 divided by 0.01, which is the same as 140 times by 100, which gives you 14,000.
Okay. Moving on to the next question now. So, what determines the osmolarity of blood?
So osmolarity is sort of the amount of fluid relative to the amount of solute. So basically the amount of fluid will be how much water there is, also determined by like ion concentrations and all the other things that are present in blood as solutes, so like glucose, ions, phosphate, calcium, urea, kind of all those things.
Okay. Next question. Why is it important to regulate this? So why is it important to regulate the osmolarity of blood?
Because, well, if there was a hypertonic solution, then fluid would kind of leave the cells. If there's a hypotonic solution, the cells would suffer from lysis. So we want to maintain that kind of perfect level of fluid where the cells don't burst, nor do the cells shrivel. And so basically we want to regulate the osmosis and the water potential and the control of that sort of stuff.
Thank you. So I believe that is the end of scenario 4, and we'll take a minute before scenario 5. Okay. So scenario 5 is about the heart. Can you describe the cardiac cycle to me, please?
So there are 3 stages to the cardiac cycle. Atrial systole, which is contraction of the atria, ventricular systole, contraction of the ventricles, and then diastole, which is relaxation. And the whole principle behind it is when the contraction occurs, there's an increase in pressure in that area, which then causes a pressure gradient. So blood moves out of there and into the next area. So go from the atria to the ventricles, from the ventricles out of the heart.
Okay, thank you. On to the next question. Can you identify what any of the sections on this ECG represent?
So the first section, the P wave, is the atrial depolarization, or atrial systole, basically. Then the PQ represents the sort of, I know that there's the delay through the AV node, which is what that is. Then the QRS is the ventricular depolarization or systole or contraction. And then T wave is relaxation of the ventricles. And I know that also the atrial relaxation is in the QRS complex, but it's kind of hidden behind the other parts of the QRS complex.
Okay. Do you mind repeating to me what you mentioned you thought the T wave was?
The ventricle relaxation. Or,
relaxation. Okay.
yeah.
Do you know another word that we would use for that?
Diastole or repolarization.
Okay. Moving on to the next question. Why is relaxation or diastole, why is that phase important?
It allows that kind of time for the chambers to fill with blood before you get another cycle passing through.
Okay. And the next question. So work out the cardiac output.
Okay, so stroke volume times heart rate. So it's 50 mils times 80, which is 4,000 milliliters per minute. So 4 liters per minute.
Okay. Moving on to the next question now. Okay. A patient comes into the hospital after sustaining an injury and losing a substantial volume of blood, so hemorrhaging. Describe the effects this will have on the circulatory system.
So they'll have a significant loss of blood volume, which means that there's less blood returning back to the heart, which then it will reduce the stroke volume and therefore the cardiac output, which can cause shock because there's less perfusion to the tissues. And in order for the body to respond to this, you'll have the sort of baroreceptors in the aortic arch that will respond to this, and they'll cause vasoconstriction, which should help to increase the heart rate and apply further stress on the heart by increasing it significantly.
Okay. The next question, how might you treat this?
So probably stopping the fluid, stopping the bleeding, sorry, first. So get some way to prevent any further fluid loss, and then replacing the fluid. So ideally using some kind of blood transfusion or like a plasma transfusion. Yeah, but in terms of first aid management, you would want to lay the patient flat, maybe even raise the part of their body that is the bleeding is coming from. And then you would want to sort of cover that up, bandage it. Yeah.
Okay. We'll just give you a few seconds before scenario 6.
I'm pretty sure I'm only doing scenarios 1 to 5, right?
That's
Guys,
true, actually. Jane doing scenario 6 to 10. That might be
Yeah,
right.
so that's totally fine. I think that's where we need to stop. So you guys finished slightly earlier, but that went pretty well.
Okay.
So I think we're going to do 6 to 10 with the next tutors. That's Jane, who'll be joining at around 7. So Hamza, if you want to go, that's totally fine.
Hi, Jane. Welcome to scenario 6, which is based on influenza. Are you ready to start?
Yes, I am. Thank you.
What can you tell me about influenza?
I can tell you that influenza is a virus, and it happens every winter, more so in the older populations. I know that there's a flu jab that people over 50 can have, or those who kind of need it on the NHS.
Okay. And what type of organism would you say influenza is?
Am I right in saying it's a virus?
Yeah.
Okay.
Apologies if you said that already. And do you know how it's transmitted?
I think normally through maybe like coughing in your hand and then touching like a door handle. Maybe the next person will then come and touch that door handle and maybe touch their face, something like that.
Okay. And as you mentioned, we have a vaccine for the flu. Do you know why we have it annually, why we need a vaccine every year?
Well, I thought that, you had a vaccine once, and then that was it, but I'm not too familiar as to why we have the vaccine annually, no.
Okay, no problem. Okay, so we'll move on to the next question.
Sure.
The viral protein that replicates the RNA is highly error prone. Why do you think this might be?
So I think this could be the case because sometimes there are mutations in the RNA, and then I suppose that kind of goes into the immune system, but I'm not too sure.
Okay. Do you know much about how our immune system, how our antibodies would respond to this?
Okay, so I know that we have something called like memory cells, where if you have an illness and then you get it the next time, you've still got those antibodies that will fight it off next time so that you're not as sick.
Okay. And now we'll move on to the next question. What causes pandemics instead of epidemics?
So I think pandemics are bigger than epidemics, but I'm not too sure of the difference between the two. I think pandemic is when like more people have an infection or an illness than an epidemic, but I'm not too sure.
Yeah, that's correct. Do you know what would cause a pandemic then, opposed to an epidemic? If it's worse.
I think it would be harder to eradicate. Is that right?
Mm-hmm. And what would you feel would be the cause of a bigger pandemic, a bigger change in the community caused by a virus?
I suppose if there were bigger changes in the genetic code, that would make it harder to overcome, and then that probably means that those who do get over the illness would have a smaller kind of there'd be a smaller pool of immunity as the virus or the infection would keep mutating.
Okay, so we'll move on to the next question now. There is current research into a universal flu vaccine. How might we develop a vaccine that protects against all influenza?
I suppose, I mean, as I said earlier, I wasn't too sure as to why we have a flu vaccine every year. But if we're going to have a vaccine that develops and protects against all influenza, I suppose we'd have to make sure that that vaccine was able to target the genome and kind of the antigens that were needed across all of the groups of influenza and possibly all the different mutations.
Okay, and this is the final one of this scenario. How might you treat influenza?
Do you mean like when you have influenza, or?
Yes, so if someone acutely becomes unwell with influenza, but also longevity-wise, how would we like to treat it?
I suppose we'd want to have drugs that could act against the antigens of the influenza. And then I suppose, as per the last question, there'd be a vaccination for the influenza that could maybe bring up herd immunity for everyone who does have it.
Well done. Okay, so we'll just pause there for a bit before we start the next scenario. Is that okay?
Yeah.
Okay, hi Jane, welcome to scenario 7. So this one is based on genes. Firstly, what is a gene?
So I know that our genome is made up of genes, and that is something that's really unique to each person. Genes themselves are made up of nucleic acids and nucleotides. For example, we have the 4 base pairs, which are A, G, T, and C. And these are made up of nitrogenous bases, a pentose sugar, and a phosphate group. Genes themselves, they code for proteins. And then one gene itself, it forms the code and then synthesizes a specific protein, for example, like the eye color.
Okay, moving on to the next question. If all cells have the same genetic information, why do different types, it's moving. Sorry. If all cells have the same genetic information, why do different types of cells have different phenotypes?
They have different phenotypes because the way that genes are expressed, it's regulated. So, for example, if I said that like gene expression affects the phenotype of a specific gene, so some of them are expressed universally, however others can vary their expression dynamically. They can also be controlled through this thing called transcription factors or more external factors like hormones, and I know that these can be reversible or irreversible. If we say, for example, stem cells, they go from being quite unspecialized to later on in their course being highly specialized cells.
Okay, on to the next question. What role do genes have in disease?
So if I give you an example, some genes are, some families have inherited mutations within genes. For example, the BRCA1, BRCA2 genes that we have with regards to breast cancer. Most disease, they do result from an interaction between genes and the environment themselves. Some of them can be genetically determined, like Down syndrome, and others can be a result from the environment, for example, poisoning. So there's different roles that genes have in disease.
Okay. Why do you think it's important to understand the genetic contribution to disease?
Well, I think there's many reasons as to why it's important to understand the genetic contribution to disease. One of them is that it's massively important for treatment in the future. For example, the same disease can be caused by different mechanisms, and this can be treated more effectively in loads of different ways. So that kind of gives a personalization to it in you know, kind of the modern world. Another point is that it's important for prevention. So if we can screen people for mutations like the BRCA1, BRCA2 genes, that later allows us to see if a certain gene or a disease will develop. And I think, you know, moving forward into the future, it helps set the foundation for the development of new treatments in the future.
Okay. The next question. How would you design a study to investigate if a gene contributes to a disease?
That's quite an interesting question. I know that we could use animals. For example, I know that in rats or mice, you can knock out certain genes that they have to see if a certain gene will do something or not. Or perhaps we could use a drug to inactivate the certain production of a gene. If I'm speaking in terms of, you know, more human-based things, we could do twin studies. So, for example, using identical twins, they have, you know, really strong traits to each other, which are influenced by genes. So we could compare these two maybe non-identical twins and see the difference. And kind of, if we do this on a larger scale, we can see if there's any common mutations between all of these people.
Okay. So that's the end of this scenario. We can take a minute now before we start on to scenario 8.
Okay.
Okay, so welcome to scenario 8. This one's on the brain. So the first question is, what is the role of the brain?
I'd say the role of the brain, there's many different roles. I suppose, you know, it's responsible for things like movement or emotions, hand-eye coordination, I suppose. But yeah, I think there's a lot of different things that can be done with the brain.
Mm-hmm. And is there anything that you feel that it does in terms of relaying information?
Yeah, I mean, it can hold information and then store it so that when we need it later, it can be used.
Okay. On to the next question. So, single-celled organisms lack a brain. How do they survive and respond to their environment?
I suppose if we have single-celled organisms, it will be a bit simpler than, let's say, human brains. So I think they'll do simpler tasks or have more simple responses. But yeah, I'm not too sure.
Okay. Moving on to the next question. How does sensory information get to the brain?
So I know the body has sensory receptors which can detect changes. I think, for example, if we speak in terms of like when you touch something hot, your brain will tell you that that thing's hot and then you won't touch it anymore. The same with pain. And I suppose just for the other senses as well.
Okay. How might painkillers selectively block pain?
Well, I think painkillers can maybe block certain pain receptors that we have in the brain or maybe on the body, which will block out the pain. I suppose maybe if you use like anesthetics, then that blocks out the pain by putting someone to sleep so that they're not in pain when a surgery happens.
Okay. And this again is the final question of this scenario. What diseases are related to the brain aging and what are their mechanisms?
I think that dementia is one of the brain diseases that you can have. I know that people who have dementia tend to not remember things and they're not usually able to take care of themselves. So that's one of them. I think maybe Parkinson's could be another one. I think that's when people, you know, tend to like shake their hands or kind of have a loss of motor function. But yeah, I think that's all that I know.
Do you know a little bit about what the main type of dementia is that would be causing the aging of the brain that we're talking about?
Is it Alzheimer's?
Yes, exactly. And do you know a little bit about the mechanisms surrounding Alzheimer's or Parkinson's, as you mentioned?
I guess it would be something about proteins and kind of maybe the receptors or not being receptive to receptors.
Okay, so that's the end of scenario 8. I'll give you a minute before scenario 9.
Okay.
Okay. So welcome to scenario 9. This one's on COVID-19. First question, why does COVID-19 cause severe disease in some people and is asymptomatic infection in others?
Yeah, so there's many reasons as to why some people are asymptomatic and some, you know, get really sick. You know, if we take into consideration the risk factors, so someone's age, maybe if they're older, they might be more susceptible to catching COVID-19. The gender, a thing that I was reading about was the ethnicity of some people. So I read that in minority ethnic groups, particularly the Pakistani and the Bangladeshi populations, they're more susceptible to catching COVID-19. I suppose, you know, the diseases can be caused by an overactive immune response, which can cause the organ failure, which is seen in severe disease. But yeah.
Moving on to the next question. Do you know any of the current drugs that we're using to treat COVID-19?
Yeah, so I heard about a drug called dexamethasone, and this is a corticosteroid which has an anti-inflammatory response and immunosuppressive effects towards COVID-19. And then I think more recently there have been trials of antibodies from some people who've recovered from COVID-19, and these are administered to people who currently have this severe infection so that they're able to obviously get better. And I suppose, I think somewhere that I read some kind of trial about vitamin D, but I'm not too sure about that one.
Okay. Okay, so we'll move on to the next question. So, how would you design a trial to test the effectiveness of these treatments?
Sure. So I know that a lot of drug trials or vaccine trials have this thing called clinical trials, and these are going from phases 0 to 4, and it has to undergo like rigorous training or kind of trials for these people. Firstly, I would randomize it in order to minimize any allocation biases and to allow equal distribution, as well as doing a blind trial so that the participants don't know what they'll be getting. And also I'd make it double blind so that there's no bias involved. I think going back to clinical trials, when they entered phase 0, it would confirm that the drug is behaving as it would be from lab research. So then we can move on to phase 1, which is a small trial of like maybe 30 people, and we're just monitoring if that's safe, if there's any side effects, which then would go on to phase 2, phase 3, increased amounts of people, start to give them placebos, start to randomize it, and then phase 4 is when the drug is licensed, and we want to learn about the long-term effects, any adverse risks, side effects, and just constantly monitor that.
Okay. Moving on to this data interpretation question. Let me know if you can see this clearly.
Yeah.
So here are the graphs produced by a randomized controlled trial investigating the effectiveness of dexamethasone in treating hospitalized COVID patients. This is a Kaplan-Meier survival curve. Describe what is shown. So I'll zoom in. Can you see A, B, C, and D?
Yeah, you can. So overall, I can see that there is lower mortality with the treatment compared to standard care. Only in the non-oxygen group, no oxygen group, is there higher rates of mortality, which obviously has the concern of potential harm to those people in that group as they lost their lives, unfortunately. I can see that each stepwise that I can see increase, it represents deaths in each group, and it looks like there's a big difference between each group.
Okay. Well done. So we'll move on to the next question now. What does R rate mean and what affects its value?
So the R rate is the reproduction rate, and what it means is that if there's one person, so let's say the R rate is 1, which is kind of the base rate. So if one person gets infected, then that one person will then transmit it onto another one person, and it's like a value. So if the R rate is more than 1, let's say it's 3, for every 1 person that gets infected, they'll then go on and infect 3 more people, and then those 3 people will infect another 3 people each, and from there it's quite like an exponential growth, which leads to things like COVID-19. If the value is less than 1, for example 0.5, then that means for each person who gets infected, 0.5 people will be infected, and so forth, until the disease eventually eradicates itself.
Okay. Okay, moving on to the next question. How could you reduce the R rate in order to slow the spread of an infectious disease?
So you need to interrupt the transmission from one person to another, and that involves isolating that person, so maybe quarantining them, using barriers to prevent any sorts of transmissions, like wearing masks, and then controlling the sort of vectors that can carry an infectious disease. You can encourage using public health interventions for people to wash their hands often, to kind of kill any infectious diseases that they may be harboring on the skin, as well as physically distancing in a society.
Okay. Is there anything else?
I suppose a big thing that I would want to look for in terms of reducing the R rate would be to look inside hospitals and make sure that there's no transmission going around there, because people who are in hospital are already immunocompromised. And so I think any kind of barriers that are put into any social setting should be amplified when it comes to a hospital setting. So as I said, for example, a big factor is humans to humans, and so you want to limit that kind of movement between, let's say, like doctors going on different wards. Doctors just need to take care of the patients that they have in order to minimize those risks.
Well done. Moving on to the final scenario now. Are you ready to go straight in?
Yeah.
Okay. So this one is on respiration. This is our final scenario. So we're starting off with an equation. Ventilation is the process by which air is moved into and out of the lungs. Ventilation is equal to respiratory rate times tidal volume. If the respiratory rate is 12 per minute and the tidal volume is 500 mL, work out the ventilation.
So I'd do the respiratory rate, which is 12 per minute, and then I'd multiply that by 500 ml, which is 0.5 liters. So I do 12 times 0.5, which gives me 6 liters a minute.
Well done. Okay. How does inspiration and expiration work?
So during inspiration, the intrathoracic volume increases, and this reduces intrapleural pressure and alveolar pressure, which then draws air in. And during expiration, the intrathoracic volume decreases, which then causes an increase in the pressure and forces air out.
Okay. How is this controlled?
So this is controlled by the brain subconsciously, but can be overridden by voluntary control. And this detects any changes in the carbon dioxide concentration and will therefore alter any ventilation accordingly.
Okay. On to the next question. How are the lungs perfused?
So The blood, it enters the circulation through the right ventricle, and then the pulmonary arteries will divide and form a capillary network which drains into pulmonary veins and from there the left side of the heart.
Okay. Below partial pressures of oxygen of 100 millimeters mercury, vasoconstriction occurs. Why might this happen?
Well, sometimes there will be blockages in parts of the bronchi, and these prevent the areas of blood from being ventilated. And also, the body, it directs blood away from these areas towards better ventilated regions through vasoconstriction.
Okay. And the final question. In what situations might this be a disadvantage, and how could you treat this?
This would be a disadvantage when, for example, the partial pressure of oxygen in the atmosphere is lower. This would then lead to vasoconstriction of every part of the lung, and therefore the blood will become quite poorly perfused. This could then lead to hypoxic pulmonary vasoconstriction, which will cause edema, which isn't great. And this can be treated using vasodilators.
Okay, lovely. Thank you very much. That concludes our scenarios for today.
Thank you.
Great job, guys.
Thanks.
So I'm just going to pause the recording there.